Hello and a very warm welcome to the final event in our 2021 season of Genome Mates, brought to you by Wellcome Connecting Science. My name is Becky Gilmore. I'm a white woman in my late 30s with short, dark blonde hair, and I'm wearing a blue top. I use the pronouns she and her. Wellcome Connecting Science's mission is to enable everyone to explore genomic science and its impact on research, health, and society. We connect researchers, health professionals, and the wider public, creating opportunities to explore genomic science and its impact on people, drawing on the groundbreaking research happening at the Wellcome Genome Campus. The Wellcome Genome Campus in Cambridgeshire is home to some of the world's leading institutes and organisations working on genomics and computational biology, including the Wellcome Sanger Institute. It's committed to delivering life-changing science with the reach, scale, and imagination to solve some of humanity's greatest challenges. Tonight, we'll be discussing how studying our genes has shaped our understanding of cancer and its possible causes. Can we predict the diseases that may affect us by looking at our genes? And what other biological, lifestyle, and environmental factors contribute to how and why we age? Since the beginning of lockdown, the Genome Gallery, our dedicated exhibition space at the Wellcome Genome Campus, connected researchers from the Wellcome Sanger Institute's Cancer, Aging and Somatic Mutations programme with artists and public participants over Zoom in a collaborative creative project called Flow Cellular. This season of Genome Mates has been part of the Flow Cellular programme, and we encourage you to visit the Genome Gallery website to explore the exhibition and the science at the heart of it. We hope that we can welcome you back to our gallery and our campus experiences before too long. But in the meantime, do check out the website and we'll put a link to it in the chat. Genomics plays a vital role in our lives and has been centre stage in recent months in the fight against COVID-19. We're now living in an age where cancer will directly affect 1 in 2 people over our lifetimes. 2021 marks 25 years since the discovery of the BRCA2 gene mutation and its link to inherited high risk of breast and other cancers. This discovery revolutionised cancer research and has a hugely positive impact on survival rates. Before we meet tonight's panel, I'd like to take a moment to tell you about the format for this evening and briefly run through some event housekeeping. Our events aim to help everyone to explore the science of genomics and what it means for all of our lives. We respectfully ask everyone to participate in a spirit of curiosity and sharing, respecting different views, identities and experiences. This platform is moderated to ensure that it's a safe and inclusive space for everybody. The panel will chat for about 50 minutes and then it will hand over to questions from you. We'd love your contributions, so don't be shy in asking. There's a Zoom Q&A box, which you'll either see at the top or bottom of your screen, depending on the device that you're watching this on. Please type your questions in there at any point through the event. You don't have to wait until you're asked or prompted. And don't worry if it looks like you're the only one asking a question. They come into a feed behind the scenes, and my colleague is working to make sure that we can cover as many of them as possible. We have closed captions available this evening if you wish to use them. To turn these on, please click on the live transcript button, which is next to the Q&A, and then choose Show Subtitles. We're delighted this evening to welcome science writer and broadcaster Kat Arney, who will be hosting the conversation. Kat is an award-winning science writer, broadcaster and public speaker. She's the author of the new book, Rebel Cell, Cancer, Evolution and the Science of Life, along with How to Code a Human and the critically acclaimed Herding Hemingway's Cats, Understanding How Our Genes Work. Kat also presents the popular Genetics Unzipped podcast for the Genetics Society, has fronted several BBC Radio 4 science documentaries, including the recent series Ingenious, looking at the story of our genes. So without further delay, I'm delighted to invite Kat and the panel to the Zoom stage to hear everyone's thoughts on what exactly is in our genes. Thank you.
Thank you very much, Becky. So yeah, my name's Dr. Kat Arney. If for any reason you're not able to see me, I am a short woman. I've got short red curly hair. And tonight I am wearing an absolutely fabulous 50s style print dress with black as the main color. And what you can't see as a bit of bonus info, it's got absolutely enormous pockets. So that's me. You can imagine me to your heart's content. So it's an absolute honor. Thank you very much to the Genome Lates team for asking me to chair this discussion. I'm really excited to dig into it because I've always been interested in how our genes make us who we are, how they affect how we develop from the first moments of life in the womb through childhood, through adulthood, and then on through life as we age. If unluckily we become unwell, how does it all work? And then in my new book, Rebel Cell, out now in paperback, it's how these changes, the changes that we inherit in our genes from our parents, and also the changes that crop up in our cells and our tissues and in our DNA as we go through life, how does that contribute to cancer? So it's a real pleasure to be joined by the panel tonight. So to very briefly introduce them, and I'll give each one a more thorough introduction later, we're joined by Professor Sir Mike Stratton. He's the director here of the Wellcome Sanger Institute. We're joined by Dr. Sandra Perez from IARC, the International Agency for Research on Cancer. And we're also joined by Maggie Blanks, who is the founder and chief executive of Pancreatic Cancer Research Fund. So first, I'm going to invite Mike onto the stage. Good evening, Mike. How are you doing?
I'm fine. Good evening, everybody. Hi, Kat.
So Mike, as I said, he's the director of the Wellcome Sanger Institute. He's the chief executive officer of the Wellcome Genome Campus, which virtually we're on. His primary research interests have been into the genetics of cancer, big old genes guy, and focusing initially on inherited susceptibility, primarily in breast cancer. And Mike was actually a key part of the team that went on to map and identify the BRCA2 gene, which, as Becky mentioned, is one of the most important high-risk genes for breast and for other cancers. And he's also played a key role in mapping other genes and genetic variations that influence the risk of other types of tumor too. So beyond just inherited gene faults in cancer, Mike initiated the Cancer Genome Project at the Wellcome Sanger Institute 20 years ago, searching for the genetic alterations in our cells as we go through life. These are called somatic mutations. And through these studies, he's discovered really important variations that influence the growth of cancers, for example, in malignant melanoma. And this has paved the way for some of the most modern targeted therapies that we have for cancer today. And the work has also led to the creation of a whole catalogue where Mike and his team are mapping the fingerprints, the signatures of different types of genetic damage in our cells and figuring out what might be causing them. And this is providing huge insights into the processes of aging and the processes of cancer. So Mike, it's absolutely wonderful to bring you on first.
Pleasure to be here.
So just to give us a bit of an overview, can you tell us a bit about what you're currently doing at the Sanger, the work that you're doing on understanding these patterns of mutation in aging and cancer? What's going on?
Well, over the last 20 years since the sequencing of the reference human genome, that's what made it possible for us, using that reference human genome as a template, to start sequencing cancer genomes, basically looking for what is different in a cancer genome than in a normal human genome. And there are two major outputs of that sort of work. The first is that genes which are mutated in cancers and which cause a normal cell to convert into a cancer cell. And that's been a mission that, the whole of the world of cancer genomics has been on for those 20 years, and it's been pretty successful. There are about 600 genes that have now been identified that, when mutated in the right way, in the right cell, they will convert or contribute to converting that cell into a cancer. And those, as you've already alluded to, Kat, those genes provide us targets for the development of new drugs. And that's not fantasy, it's not rhetoric. There have been many new drugs that have been developed using those mutated genes as targets, and those drugs work. So that's the first major output of sequencing cells for so-called somatic mutations, the mutations that take place throughout our life in all cells of our body. The second thing is in the cancers, what are the patterns of mutations that we see? Because we now know that there are many different mutational processes, factors, processes, biological conditions, exposures, lifestyle, that can change which mutations are being generated. And so, for example, looking in cancers, in lung cancers from smokers, you can see very distinct patterns of mutations that we now know are associated with or due to the chemicals that are present within tobacco smoke. Similarly, but a completely different pattern of mutations you can find in malignant melanomas and other tumors of the skin, which have been caused essentially by ultraviolet light, and ultraviolet light also causes mutations. So the same overall process, but two completely different patterns, such that you can give us blind the sequence of a lung cancer or the sequence of a malignant melanoma and not tell us which is which, and we'll be able to say which is which, because one contains the mutations typical of ultraviolet light, the other contains the mutations typical of carcinogens in tobacco smoke. So we've now been continuing that to look for all the mutational signatures and to really dig deep into cancers and now into normal cells for all the mutational processes that are afflicting us, that are causing mutations. Some of those are external, they're exogenous, they're due to exposures, but actually we generate a lot of mutations ourselves just by being alive. It's a natural process.
So one of the things that I found most interesting when I was researching my book was the discovery that, you know, we think of genetic mutations as only being bad and only being found in cancer cells, but there's so much of our normal tissue has these somatic mutations, and they may or they may not actually be important, that we're full of mutations all the time, but most of them aren't bad. So how does that sort of connect through to then the discovery that there are certain mutations that actually will then drive a cell into becoming a cancer? And is it just one gene or many genes? So what's the relationship between all these mutations and then what makes a cancer cell, a sad cell become a bad cancer cell?
Right, well, there is a partial answer to that question, but there are still big mysteries. So what seems to be true, and this comes from some of the most recent work that we're doing at Sanger, by looking at normal tissues, that normal tissues are acquiring mutations the whole time. You may think it peculiar that work on normal cells came after doing the work on cancers, but there are good technical reasons why actually sequencing the genomes of normal cells is much, much more difficult than sequencing the genomes of cancers. And so until very recently, we knew much more about the genomes and mutations present in cancers than we did about normal cells. But now we've got a pretty good, still evolving picture of the mutations in normal cells, and basically all normal cells in the body throughout life accumulate mutations. But the fact is that the vast majority of these are in every cell are completely innocuous. The huge overwhelming majority are. And a cancer arises when a mutation falls completely by chance in the correct gene, and it has to be the correct type of mutation as well, that then converts that gene into telling the cell to go down the path to becoming a cancer. And the reason I use that rather elaborate wordage is because a mutation of one cancer gene is simply not enough to convert a normal cell into a cancer cell. Indeed, and we've sort of known this for a while, because when we look at cancers themselves, we find 2, 3, 4, 5 mutated cancer genes in each cancer. So obviously you require that many mutated cancer genes to get there. What has been a little bit more surprising actually has been looking in normal tissues over the last 3-4 years and finding that actually we find cells, little clones of cells, microscopic clones of cells that actually look normal under the microscope. You can't distinguish them by looking at them, that already have a mutation in a cancer gene.
This is so interesting, this observation.
Yeah, and those clones are expanding. They're growing faster than a normal cell that does not have a cancer gene in, but still they look normal under the microscope. And such a clone can have one mutated cancer gene, but we've also found clones that have 2, clones that have 3 or even 5 mutated cancer genes, and yet down the microscope, they look completely normal. So you need a cell needs to cross a lot of hurdles. All of these different mutated cancer genes before it gets enough disruption of the cellular physiology that then causes it to become a cancer.
And this is something we'll probably come to a bit later when we have a more broader discussion, but it's that interesting question of, even if you have these cells that are poised to go rogue, it's not enough just to have the mutations. Cancer is not just about this accumulation of a shopping list of mutations and then you're off. There's sort of a broader biology at work, I guess, because these cells are in our tissues, in our bodies, and they're subject to all the sort of pressures and life as we're living it.
Yeah, well, there's certainly the finding of these clones of cells with cancer genes mutated and often multiple has put that particular question right up there to be answered. Yeah, it could be that there are other things happening in the biology of cells, some things that perhaps we sort of know about, like epigenetic changes, not actually mutations, but changes in the genome structure that can be transferred from cell to cell, that those could also be contributing. But some of us, old-timer cancer geneticists feel that we want to push the boundaries of the mutations hypothesis as far as it goes. And I'm not completely convinced yet that the right set of mutations and the right set of genes, and in fact, disorder of the genome, ongoing elevated rates of mutation are actually enough on their own to take the cell all the way to that cancer phenotype.
I think this is probably an argument we can take offline. Yeah, so one thing that I did want to tie in a bit is obviously from your work now 2 decades ago, discovering that changes in the BRCA2 gene are associated with strongly inherited patterns of breast cancer, and then discovering ovarian cancer and prostate cancers that are all associated with these inherited changes in the gene, and then trying to tie that through to what we now understand about these somatic mutations, the changes in our genes that we pick up through life, and sort of how do we thread those two together? Because it seems to be two almost completely parallel strands. We've got inherited genetic variations, and then we've got somatic variations. So how do we match those up when it comes to understanding what's driving cancer?
Right. Well, they're both part essentially of the same story, and the story is that for a cell to become a cancer cell, you need multiple key mutations. Now, in the case of BRCA2, and somebody who's at elevated risk of breast and ovarian cancer because of an inherited mutation of BRCA2, every cell in their body has got one of the 2 copies of BRCA2 mutated. But the other copy is perfectly normal. And that other copy that's perfectly normal keeps all the cells in the body going perfectly well. Cells only need one functioning copy of BRCA2. However, we've just been discussing that mutations take place all the way throughout life in all cells of our body, and there are all sorts of different types of mutation. And if in a single cell, the second copy of BRCA2, the normal copy, gets mutated, now you've got a cell in which both copies are mutated, and there is no longer the function provided to the cell that BRCA2 should be giving. And that's a cell that now may go on to become a cancer. But the word may is key there, because we believe that even that is not enough for a cell to become, for a breast cell to become a cancer cell. Because actually, if both copies of BRCA2 are mutated, that's a pretty toxic insult to a cell. Basically, a key component of DNA repair has gone, and the genome of the cell begins to break up in all sorts of chaotic ways. And cells don't like that, and cells have got mechanisms to deal with that, basically sending the cell into death. So the cell dies. So you don't get a cancer from that cell. So we think that probably in the case of BRCA2 and BRCA1 and some other genes similar to that, what happens is you need yet another mutation in a gene that is involved in those pathways that send cells into death. And if you've inactivated, if you've made dysfunctional or non-functional that pathway, then a cell which has got both of its BRCA2 copies mutated now can produce that chaos in the genome of the cell, but the cell won't die. It'll just continue accumulating mutations, and that's absolutely the recipe you need to generate a cancer.
So one of the curious observations, I have a friend who has, she's inherited a mutation in her BRCA2 gene and went on to get breast cancer. But as she pointed out to me, she's like, every cell in her body carries this mutation that she's inherited, but she's only at increased risk of breast and ovarian cancer. So, you know, what do we know about the tissues in which cancers arise? Because we know as well, for example, in the case of malignant melanoma, you know, you get melanomas that are more likely to have changes in this certain gene called BRAF, which you were also involved in discovering. So what do we know so far about why different cancers arise in different tissues?
Well, it's, for the most part, it's something of a mystery, actually. Why individuals with BRCA2 inherited mutations get breast and ovarian cancer, and the same thing for BRCA1, is really not known. It's been speculated on for many years. A simple proposal would be, well, you know, maybe that's the tissue in which BRCA1 and BRCA2, breast and ovarian tissues, that's where the gene is expressed. That's not true. It's expressed all over the place. And so we actually don't really have a good answer to that question for BRCA1 and 2. We don't really have a good answer to it for an awful lot of the cancer predisposition syndromes. For example, there are cancer predisposition syndromes that are caused by causing an elevation of or disruption of a different DNA repair process, or so-called DNA mismatch repair. And those individuals get colorectal and endometrial cancer. And you could imagine that, well, maybe this repair pathway is particularly involved when cells divide, and that's the reason, but all cells divide, basically. So why is it that we just get colorectal cancer and not colorectal and endometrial cancer? Actually, we don't really know. And there is, some of these mysteries are the best ones to sort of chew on, are the oldest. And whether we're talking about cancer predisposition or we're talking about the general population incidence rates of cancer, one of the observations that was made long time ago was that actually colon cancer is quite common, so that's the large intestine, but cancer of the small intestine is incredibly rare, right? And these things join one to the other. Why should that be? Well, is there more tissue of the colon than small intestine? Absolutely not. There's probably more tissue of the small intestine, so that's not an explanation. And so, of course, in times of, you know, pleasurable ignorance, we could speculate and we could say, well, maybe what it is that the mutation rate generally is higher in the colon than in the small intestine. Well, as I've been discussing with you, we've been able to look at normal tissues in the last 2 or 3 years, and that is absolutely not the explanation. The mutation rate in colon is exactly the same in normal colon as in normal small intestine. So differences in the mutation rate are really not at the bottom of these very dramatic differences in cancer incidence. So there are these really key mysteries that were cited and proposed to us decades, centuries ago even, that we still are finding difficult to unravel.
It's fascinating. I know that we have some A-level students watching us tonight, so maybe that's something to get your teeth into when you go on to university. I think would be a fantastic problem that definitely does need solving. Thanks very much, Mike. We will come back to you when we have the panel discussion. So the next person that we're going to be speaking to is Dr. Sandra Perez. She is a genetic epidemiologist who works as a scientist in the genomic epidemiology branch at IARC, that's the International Agency for Research on Cancer in Lyon. And she's worked there since November 2019. Her research focuses on using genomics, that's understanding our DNA and our genomes, to understand the major risk factors associated with cancer in different parts of the world, because cancer rates are different in different places. She's built international collaborations on head and neck and breast cancer with research groups mainly in North America, Europe, and in Latin America, combining large population and patient group studies with really innovative lab research. So, hi, Sandra, do you want to pop onto my stage?
Hi, Kate. Hi, everyone. Maybe for those who are not seeing me, I'm 41 years old. I'm originally from Colombia, so I identify myself as Latin, and I'm wearing a blue shirt.
Fabulous. And you have a very beautiful painting behind you, which looks kind of like a very abstract, colorful scene. So
yeah, it's a scene of a central market in Bogota.
Absolutely fantastic. So Sandra, can we start off by talking a bit about what you do at IARC? You know, what we know a bit, maybe a geneticist is someone who studies genes. What's an epidemiologist? You study epidemes? What do you do?
No, actually, epidemiology is a common word now after the pandemic. I mean, we work at the genomic epidemiology section, which I think that it identifies really well what we do. We combine those two areas of research, the genomic part with different genomic techniques and type of analysis. The epidemiological part comes with the design and the collaborations that we have with the studies all over the world. We really want to combine these two areas of research to try to understand the natural history of cancer, to try to understand the different steps that lead to cancer development. We want to know, for example, what are the causes of cancer? And if it's only our genetic background, is it exposure to lifestyle factors? We want to understand especially those factors that could be preventable, because then we will have some strategies to try to prevent the development of cancer. But we also are interested once the cancer develops, we want to know if there is a specific alteration or if there is specific exposure that we can use to try to improve early detection of cancer. And after cancer develops, we also want to understand why some cancer patients will have a better outcome after treatment compared to others, or if there is any specific response to treatment based on the genomics and also based on this specific lifestyle factors that the person had to during their lives. So the epidemiological part of our studies comes from the international nature of the agency where we work. We have a lot of collaborations with many centers in different parts of the world. We designed and coordinate large studies in which we recruit cancer patients. Depending on the type of a study, we also recruit healthy individuals. We collect a large amount of data, lifestyle data, demographics, we collect clinical data, we collect a lot of biological samples because we're interested, and we have a specific focus on low and middle income countries. I mean, we have collaborations all over the world, but we have a specific focus on low and middle income countries.
So I was wondering if you could actually give me some examples of how the patterns of cancer are different in some of these places, because I know here, you know, in the UK, our top cancers are lung, breast, prostate, bowel. So how do the sort of the prevalences of different types of cancer? Can you give some examples of where you see these incidences that are different in different places?
Yeah, that's actually one of the questions, the key questions that we want to understand. And it's one of the key questions that it's a question that always appears when saying, well, why this cancer is specific of this population and why this other is more frequent in another population. And we see some difference even in the same region or even in the same country, in different parts of the country. There are,
wow.
the frequency differs a lot. For some of the cancers, we know what the answer is, because we know the etiology, I mean, the cause of cancer behind the development of that cancer. One important and very straightforward example is cervical cancer. We know that most of cervical cancer are associated with HPV infection. So we know that the prevalence of HPV infection before the vaccine, if it's very high, well, we understand why this is the most important type of cancer in some countries. But for some other cancers, we really don't know. I mean, the causes that are associated with those cancers do not really fully explain why these cancers are more prevalent in one population and another. One example of that is colorectal, in which we know that there are some lifestyle factors that are associated with the higher frequency of colorectal cancer, like obesity, but it's not enough. I mean, it's not enough to try to explain why we see these high incidence cases in, for example, North America compared to other countries in which the incidence is lower. So this is one example of why we are working, especially with the team in Sanger and with Mike and Maggie, in terms of understanding using genomics, why we see these differences in incidence, and if we can tease out a little bit if we can find another cause or possible lifestyle that could be also associated to the increase of cases for specific cancer types. So
this is the work that you're doing with the Mutagraphs of Cancer project. So this is a big grand challenge project funded by Cancer Research UK, and I've been really lucky to be involved in reporting on some of this. And tell me a bit about actually what this project is and what you're trying to do, because it's thousands of cancer samples from thousands of patients all over the world with different types of cancer. What are you doing? What are you looking for? And what are you hoping to understand?
We're actually hoping to understand the question that we were just discussing. I mean, why we see these differences in incidence in specific cancer types that cannot be explained by the known risk factors. So what we're doing now is that we're collecting 5,000 or more than 5,000 cancer cases all over the world. We selected 5 cancer types for which we know that there are specific differences in different regions of the world. That is kidney cancer, colorectal cancer, pancreatic cancer, esophageal squamous cell carcinoma, which is one type of esophageal cancer, and the other type of esophageal cancer, that is esophageal adenocarcinoma. So for these 5 different cancer types, we have collected, or we have recruited cancer cases in different parts, depending on the incidence of that cancer type in different regions of the world. And we're looking at the mutational signatures, the mutational patterns that Mike previously discussed and described. And we want to compare the differences, how this mutational signature in the colorectal cancer of a case that is in Canada compared to the mutational signatures of a colorectal cancer case in Thailand, for example. So we want to see looking at these differences in mutational signatures, we can discover probably a different cause of cancer, or maybe we can kind of see different mechanisms of cancer development that might explain somehow the differences in cancer cases into regions.
The thing I love about this project is often we talk about, the causes of cancer, and maybe it's things in the environment or things in our lifestyle or things we don't even know about. And we can prevent the cancer by avoiding these exposures, but then you need to know, what is it? What are the things, and how are they actually causing cancer? This is real prevention research. And so what can we learn then, and what can we do with this information once we start to piece together there are these cancers and these genetic mutation patterns and these risk factors? How do you sort of crunch it all together?
Well, one important message here is the diversity of cancer patients that we are analyzing, for example. I think this is something to highlight about this project, because we are looking at, for example, the specific exposures that are specific for some regions and not all over the world. And sometimes are exposures that we didn't know much about it. We didn't know that they were linked to a specific cancer type. That's the case, for example, of opium consumption and in certain populations in Asia. And we want, I mean, we really need to understand what is causing the cancer, and if there is a specific exposure for which at this moment we don't have sufficient evidence to try to say it can be carcinogenic, then with this project we have sufficient evidence to try to say, well, we have to pay attention to this specific exposure because it may cause cancer. So that's one thing. But also we need to understand the interaction between these exposures and our background and the lifestyle of people who are actually we are studying. So that's also interesting. We have to be careful also as well to try to kind of communicate these results, because as long as we are just giving kind of the first line of evidence of a possible exposure that could be linked to the cancer, to the development of cancer, but we obviously will need to accumulate a lot more evidence to try to say, yes, this is something that we have to pay attention and probably to try to translate it to, for example, preventive strategies. So we are just in the beginning of the first step to try to understand the different causes or new causes of cancer.
Thank you very much. So we'll come back to some of perhaps these challenges of how do we communicate these messages when we have a discussion later on. But thanks very much for now, Sandra. And so next, I would like to invite Maggie Blanks onto my virtual stage. So hi, Maggie.
Hi.
Do you want to briefly introduce yourself?
Yes, hello. My name is Maggie Blanks. I think I'm Caucasian. I'm not divulging my age, but the fact that
a lady need not.
The fact that I've got silver hair might give some ballpark indication.
Thank you. And you're wearing a very fetching stripy blue shirt and have some lovely, are they roses? Yellow roses in your background. Roses. Yes. Beautiful. So Maggie, as well as having a very attractive shirt and backdrop, is the founder and chief executive of the charity Pancreatic Cancer Research Fund, set up in 2004 to support research after the death of her husband from pancreatic cancer. And I know that this is really absolutely a driven passion for you. And having spoken to you about this, your work is really fantastic. Maggie is currently a patient advocate involved in Cancer Research UK's Grand Challenge Mutagraphs project, and it has been my pleasure to travel with her to Kenya and also to the Czech Republic to report on cancer patients' experiences in this country. So Maggie, let's start off. If you can give us a brief overview of what do you actually do at the Pancreatic Cancer Research Fund? Because I think, it's a cancer charity, good thing. How does that work? What do you do?
Everything. Yeah, as you say, Kat, I set up the charity in 2004. Gosh, haven't the years flown? After my husband's death from pancreatic cancer. And it wasn't a sort of an automatic reaction after his death to start a charity, because heaven knows there are lots and lots and lots of charities in the UK already. But what the trigger was discovering two things, I guess, after Alan died, aged 56, I have to say, from pancreatic cancer. One was my discovering that the then survival rate, 5-year survival rate for pancreatic cancer, not only was abysmally low then, it was about 3%, but that it had been the same for decades. And the other thing I discovered at that time, just after his death, was that the amount of research funding going into pancreatic cancer specifically from the major funders in the UK, well, and elsewhere, certainly in the States as well, the amount of research funding for this particular cancer was also abysmal. It was tiny. And although I'm no scientist, and I didn't know anything about kind of cancer research at that time, it was very obvious from seeing the improvement in survivals for other cancer types that investing in research was damn important. It was key to change. And I thought, well, then I must do something to help get more research done into pancreatic cancer so that things could be more hopeful for future patients. It was too late for my husband, Alan, but I thought if something good could come from doing something in his name, then that's what I was going to do.
And what kinds of research do you fund through the fund? Because obviously I can see that there's a desperate need to improve survival through treatments, but also I guess a desperate need to understand what's causing these cancers as well.
Yes, certainly we're a small charities then and still are. And the way that we fund research is through awarding research grants. We have a peer review process like all good medical research charities. We have one award round a year, and it's a competitive process, and the applications for grants that we receive are reviewed by our scientific panel and external reviewers. The kind of research that we fund, certainly in the early days of the charity, was the fundamental science, the base, what also called basic science. Because what I learned and continue to learn is that in order to make progress in tackling any cancer, you have to understand the biology of that particular cancer. Each cancer type has its own kind of biology, and there are important questions about, you know, how it operates, its mechanisms, and so on and so forth. And the understanding of that biology is essential before you know what you're dealing with and what particular targets there might be, to the types of treatment that might be relevant, and so on and so forth. So in the early years, given that there had been very little research into pancreatic cancer before 2004, the kind of applications that we were receiving and funding were the basic science. So it's the lab-based type of research. As the years have gone by, and there has been a growing understanding about the biology of pancreatic cancer, the kind of mechanisms that make it the aggressive cancer it is, how it spreads, why it resists, why it's so good at resisting the chemo treatments that can be effective in other cancers. As that understanding of the biology has widened, then we seem to be doing more of a combination of two types, I guess, of research. Still some of the basic science stuff, but also what's called more translational research. So it's not yet at the clinical trial stage, because that costs a lot more money than we've probably got.
Oh yeah.
but translating some of the knowledge from the basic fundamental science into something that you can test in an animal model rather than just look at cells in a dish. That's the translational stage. So it's moving forward a hypothesis about how a particular drug or something might work, but it has to be tested in something like mouse models, some kind of, you know, organism, in order to see how the thing operates in something that's living.
I wanted to explore a bit about your work as a patient advocate, and also just what the families and people with pancreatic cancer want to know about, because the connection to the BRCA2 gene is that having inherited a fault in that gene actually does put some people at risk of pancreatic cancer as well. And so, you know, how have things changed for patients, people with pancreatic cancer in understanding what actually is causing this disease? Because it is a question that everyone asks, you know, when you're diagnosed with cancer, is why me? Why did this happen?
Yes, why? Yes, yeah, that's absolutely true. But the majority of pancreatic cancer cases that are diagnosed each year are sporadic. That means they're a one-off. It's bad luck, but it's a one-off. It seems that only around 10% of the pancreatic cancer cases diagnosed each year are in families where there is a family history of pancreatic cancer. And so while the involvement of those families in research to try and understand more about the hereditary elements, what are the genes predisposing the members of that family to pancreatic cancer, for the majority of pancreatic cancer patients, the challenge is still to understand what triggered their particular cancer and how best to fight it.
And have attitudes changed as well over the time? I remember when we were preparing for this, you told me a story about, you know, the sort of the changing attitudes of doctors towards patients with this cancer.
Yes, I was quite shocked to hear from some of our supporter families in the early days of the charity anyway, the stories that in the past, say 50 years ago, when cancer generally had more of a stigma attached to it. You know, it was not even, the word wasn't even kind of used much, was it? It was the big C, and people, some of these families said that, yeah, where they'd lost people to pancreatic cancer in those days, the assumption, the general assumption on the part of many medical professionals was too much drink, that it was it was the drunks who got pancreatic cancer. Yeah, we know different now, but
yeah, I mean, this just shows how important all this kind of research that's going on is to actually understand what is causing these cancers rather than just making assumptions about it. Thank you very much, Maggie. I'm going to invite Sandra and Mike to come back on the stage now. We're going to have about 10 minutes of discussion. I've got some questions, and then we're going to go to the audience questions, which I'm sure are better than my questions. So I'm going to start with Mike, actually, because this is something that I wanted to ask you. We ran out of time in our chat, and then also Sandra as well. So from Mike, all the research that that's ongoing into understanding these kind of genetic changes that are driving cancer, the inherited changes involved in cancer, how can we actually use this to improve treatment? And then I'll come to you, Sandra, and ask about how can we use this to improve prevention, if that's all right. So yeah, Mike, how can we turn this information into things that will actually benefit people being diagnosed with cancer now or in the near future?
Well, in the first instance, when the knowledge that when you discover a mutated gene that is clearly causally implicated in converting a normal cell into a cancer cell, that screams at you that if you could somehow tamper with the function of that gene, you could perhaps revert that cancer cell to being a normal cell. And there are a subset of genes that what happens to them when they're mutated and they contribute to the causation of cancer is they're switched on. That's what they are done. Instead of being switched on and then switched off in a completely regulated way, they're stuck in the on position by the mutation. That's what the mutation is doing. And so the mutation in that gene is duping the cell. It's telling the cell to keep on growing, which is, after all, what a cancer is. It's a cell that refuses to stop growing when it should do. And therefore, it's a very simple step from there to say, well, if we could develop a drug that could somehow get into the protein made by these mutated gene of that nature and somehow shut it down, then the signal to those cells, because there'll be billions of cells now in the cancer, to keep on growing will disappear. And so the cancer might stop growing. And that was a notion that was right there from the outset of the search for these mutated cancer genes as to what they could deliver. And we now have multiple examples of this where it has worked. So one of those that you mentioned previously that I was involved in was with respect to the BRAF gene. And we did a completely blind screen, actually, through a set of cancers in the very early days of sequencing cancer genomes. It was a mixed set of cancers. We chose the genes in a completely ad hoc way. And one of those genes, I remember very clearly the day, started spitting out mutations on our sequencers, and that was BRAF. Mutation after mutation started coming out. And these mutations, we could put them onto our map of the protein, and we could see pretty clearly that these were the sorts of mutations that would be switching the protein on and keeping it switched on. So we published that, and immediately multiple pharmaceutical companies started developing drugs against the BRAF, the mutated BRAF protein. And those were developed really pretty quickly, actually. They were put through clinical trials really pretty quickly. And, you know, I, again, can remember very clearly the first pictures that we were seeing. And these were pictures not so much of, you know, the patients beginning to get better, but scans of their cancers. So these were patients with disseminated malignant melanoma, really widely spread. The PET scans in particular showed the activity of these cancer cells. After a couple of doses of the BRAF inhibitors, they just shut down. It was like a light going off.
It's an incredibly powerful demonstration of when it works to turn this genetic information into treatments. And then on the prevention side, Sandra, when this kind of information, we talked about it a bit, but, you know, when we find information about what does link risk factors, whether that's in the environment or lifestyle, to cancer, what can we then do with that information? Is it just for us to worry about? Is it just for the government? How do we turn this information into prevention strategies?
I think the answer would depend on the exposure, the lifestyle factor. I mean, we know it very well for smoking, for example. We know that smoking causes a large number of cancers, especially lung cancer, and it has a mutational signature or mutation pattern that is linked directly to smoking. And we know all the possible strategies that we can use to try to prevent exposure and obviously to prevent development of cancer due to smoking. Is not the case for all the exposures or all the lifestyle factors. For example, we know that the second lifestyle factor now that is causing cancer is obesity, but we do not have a signature of obesity in the genome. So there must be something else. There must be something else that is doing obesity, and that obesity is doing in terms of causing cancer. I think for those causes for which we know that there are preventive interventions, we can do more as an individual, but unfortunately we don't live isolated as individuals. We live in a society. So as individuals, we can obviously try to change behaviors and try to prevent exposure. But since we live in a society, this is where the public health policies play an important role. And as a society, we have to invest in having healthy people and happy people. Healthy people are productive people as well.
And happy people, generally.
So this is something that has to be communicated directly to the policymakers, saying you want to invest in a better society, you have to invest in the health of your people. And if you have information about possible causes of cancer that can be prevented, then you have to invest in the best strategies to prevent that exposure.
And Maggie, I wanted to bring you in here because there's a slightly difficult needle to thread. And I found this when I've been writing about cancer risks and causes, because you're aware that, you know, you can tell a policymaker, oh, we know these things are risk factors and we should do things about it. But as a patient or someone from a family of someone with cancer, it can sometimes hear like, well, you're hearing these things and are you blaming people? You're saying, well, if you hadn't done that, or, you know, you did this. So how do we thread this needle of messaging? What do you see as a way through?
I'm not sure I do at the moment anyway. It is difficult. Where we're talking about, say, pancreatic cancer, for example, as Mike was saying earlier, when the question is asked, well, what's caused this? Some of the recent research statistics suggest that in about 20% of pancreatic cancer cases, smoking or excessive drinking might be implicated in the cause. But what about the rest? And I often think of a young man of the youngest patient I know who died of pancreatic cancer, a young man of 29, was a serious athlete, and therefore his lifestyle was exemplary in terms of, you know, what he ate and how he looked after himself and so on, and died of pancreatic cancer. So the messaging is not self-evident, I think, at the moment. Other than we all know we should, you know, have a good diet and take regular exercise and so on and so forth. But I know from talking to many bereaved families to do with the charity that lots of people have died of pancreatic cancer who exactly fit that description.
You know, it's the thing, it's like, well, my granddad smoked till he was 90 and he was fine. I'm like, well, my granny smoked and died in her late 60s. So, you know, so Mike, sort of bring you in a bit here. What is this balance then between mutations and environmental factors that mean that some people who smoke don't get cancer, some people who don't smoke do get cancer? This, you know, it's not a given, is it? It's not a, if you do this, then this.
No, absolutely not. None of these absolutely definite risk factors ever confer a certainty of developing cancer. They will increase the risk to varying extents depending on the risk factor. And therefore there will be people who did get cancer as a result of whatever that exposure is, tobacco smoke, and there will be people who didn't. There is always in the core of the process, there is if we just think about it in mutations, it's in the core of the process, there is an element of chance. And you can imagine a mutation landing in the right place in a cancer gene and causing that cancer gene to convert a normal cell to a cancer cell. If that mutation was in the letter of code next door, it wouldn't do that job. So that's the sort of chance that one of the elements of chance, if you like, that's occurring as these exposures contribute to their elevated cancer risks. And similarly, sorry, you go.
No, carry on.
Well, and similarly for what we were talking about previously, the risks of breast and ovarian and pancreatic cancer and prostate cancer in BRCA1 and 2 carriers, carrying the mutation, the predisposing mutation, every cell of the body does not confer a certainty of developing the disease. It's a high risk, and it's a much higher risk than in the general population. But clearly, there is a subset of people carrying those mutations that can get through life. Some of that may be due to chance. Some of that may be due to so-called modifying genetic elements that together can sort of reduce the risk, or in some people probably increase it. And in some, probably there are elements of lifestyle which are also modulating the risk. So it's a complex final equation that has to run its course to generate that risk of cancer.
So as I've been going out and doing a lot of talks about my book, which is all about, you know, how does cancer start? How does it evolve? How does it change in the body? People have asked me, oh, what's the best way of preventing cancer? And I always think, well, one, don't add to your mutational burden. You know, don't do things that we know add mutations. And then 2, like, just do the boring stuff that we know keeps you healthy. I don't know, Sandra, are there any more salient points that we can add there?
If you're selling us a boring thing, no one is going to do it.
Exercise is fun. I love it.
Exactly. Yeah,
Dancing.
I think so. I mean, at some point, I mean, cancer, as any chronic disease, is a disease of aging. And this is something that we cannot modify. We age, and that's we have to live with it. And I do agree. I mean, the only thing that we can do is for those specific risk factors that we know that are associated to cancer, well, that we can prevent, not to be exposed to, or reduce the exposure, and do all the things that we know that could prevent cancer, like have a healthy lifestyle and have a healthy weight and exercise in a fun way. I mean, there are different things of exercising, fun things to do. But then, I mean, there are things that you have to keep living. I mean, there must be a balance. I mean, you have to have fun, but at the same time, you have to keep healthy, because if you're not healthy, you are not going to be able to enjoy the things that you like. So this is something that you have to consider.
Absolutely. We're going to turn now to the audience questions. It's lovely to see them coming in. If there's more questions that you think of, please pop them in the Q&A, because that's how we'll see them. If you pop things in the chat, we're not going to see them. So top question, first of all, and this I think is a great question. We'll start with you for Mike, with you for this one. Why would nature give us these cancer genes? You know, why are they in our bodies? Is there any advantage to having mutations? So these seem like very risky genes to have if they can become cancer genes. Why have we got them?
Well, I think there are 2 questions in there. Why do we have these cancer genes, which when mutated can cause cancer, and why do we have mutations at all? So why do we have mutations? Well, we have to have mutations because if we didn't have mutations, none of us would be here. Life has evolved because DNA replication, and in the very early days, RNA replication, is not completely perfect. Mistakes get made, and because of those mistakes getting made, organisms can explore all sorts of environmental niches because their genes are being changed and they can accommodate. So mutations are absolutely at the bottom of evolution, and those mutations need to continue as organisms because they're part of our makeup. So then why do we have those genes? Well, because those genes do a pretty good job day in, day out. You know, the process of us developing from a single fertilized egg into an organism of trillions of cells, and then to maintain all of this over 70 plus or whatever years of lifespan, that means that it has to be incredibly well coordinated. Cells have to be told to divide at this moment, not to divide at that moment, this cell to divide, that cell not to divide. It's an incredibly complex process which requires an incredibly complex machinery, which when well regulated, does a wonderful job, an extraordinary job, almost an unimaginably extraordinary job. It's when in the occasional cell, it goes wrong. That's when the cell gets out of control, and that's when a cancer occurs.
We've got a question here from Ignacia Arteaga. I expect a fellow Spanish speaker, I think. I wonder if speakers, especially Sandra, could speak about the impact of climate change in increased carcinogenic exposure and prevalence. Do we observe in the epidemiological studies any changes there that we can tie in?
Possibly the more direct link between climate change and exposure will be the influence of food availability. I mean, the influence of climate change in crops, obviously. And nutrition is one of the important factors that could modify, actually, it could be a risk factor depending on the type of food that you're consuming, or a protective factor for specific cancer types. And it's also obviously linked to having a healthy weight and preventing obesity. So I think, yes, I mean, for climate change has an important impact. It's not only the consequence of practices in culturing food, but also it's a direct cause of changing sometimes dietary patterns in many populations. And this has a direct impact in what we consume and the possible risk factors that are increasing for specific cancer types.
So one to watch, I guess.
Yeah.
Here's a question. I'm going to put this to you first, Maggie, because this is a question from Kevin, who asked, to what extent are the poor survival rates in some cancers, such as pancreatic, a result of late diagnosis rather than our ability to treat them when found? And I know these things are sort of closely tied. So do you want us to talk us through the challenges of late diagnosis?
Yes, Kevin is absolutely right that certainly about pancreatic cancer, that the poor survival rate is very much a factor of the late diagnoses, and it is a very difficult cancer to diagnose early. In its very early stages, there are often no symptoms. So someone can be developing a pancreas tumor and feel pretty much their normal cells. And later, as symptoms are appearing, the symptoms are often vague, non-specific, not consistent from one patient to another. Vague, like, for example, some people can get what feels like indigestion. So you get the situation of people self-medicating with, various bottles of stuff or tablets for quite a while before things get so bad that they worry that it's something else. So those factors end up in people, by the time they are aware of something seriously wrong and get diagnosed, by then the disease is pretty advanced. And because pancreatic cancer is very good at spreading, metastasizing beyond the pancreas to other organs, that generally rules out surgery, which is at the moment the only potentially curative treatment. And so this dire survival rate is, yes, very much a factor of late diagnosis. And I think early detection is a big hot topic now for most cancers, many cancers, but it's certainly a priority area for pancreatic cancer.
So it feels like that's a whole separate topic for a whole separate event. A fantastic question has come in from Jay, and Sandra, I'm going to put this one to you because I think it's a great and very clever question. Does the epidemiological research highlight areas where certain cancers are of lower incidence? So this would be good news and could help us to understand what's protective against cancer. So rather than always looking for what's gone wrong, can we look at what doesn't happen? The dog that doesn't bark.
Yeah, wonderful. I really like this question because it is true that we focus a lot on talking about risk factors and what is causing cancer. And we need to probably back up and say, well, we need to look at what is protective and also motivate people to try to start doing this. Yes. We have a lot of, known lifestyle factors that we know now that are protective. Associated with, for example, in nutrition factors, the consumption of fruits and vegetables and fiber, rich food rich on fiber, physical activity as well. We know that for many of the epidemiological studies, the benefit of physical activity to preventing cancer.
So one area I thought was very interesting that I discovered while researching my book was that we know that the rate as you get older, particularly past 60, your incidence of cancer goes up. But then if you can get to like your late 90s, it actually goes down. So like if you've made it to old age without getting cancer, you're probably not going to. So I find like trying to study the very oldest old and what's going on, and that presumably might well be something in their genes. Would you agree?
Yeah, definitely. And it's not only, as we've said in the whole session, it's not only the lifestyle, it's not only the genes, it's the combination of both areas. So we need to look at both at the same time. That's definitely true.
It's the nature and the nurture. Just quickly scrolling through our questions here. So Mike, here's a question for you. So you mentioned the gene-based drug that, for example, blocks BRAF when the BRAF gene is overactive in certain types of melanoma. Why does that not work across all types of cancers? So if you could sort of explain a bit like, well, why isn't BRAF a treatment for all types of cancer?
So the first reason is that BRAF mutations are only found in a certain subset of cancer. So they're found in about 60–70% of malignant melanomas, but they're found in a very trivial percentage of breast cancers. So treatments for BRAF can really only work in malignant melanoma and other cancer types. Colorectal has about a 10–15% chance of having a BRAF mutation. The drugs that are developed only work in cancers with BRAF mutations. If there's not a BRAF mutation there, the drug won't work. So it works in that 60% of malignant melanomas, but not in the other 40% that don't have the mutation. And it won't work in other cancer types which don't have BRAF mutations at all. The question more generally of why, given the fact that we believe all cancers need to have mutated cancer genes, don't we have more drugs that just treat them all? And the answer there is that certain genes lend themselves to being drugged. So, for example, and BRAF is one of those genes. So when we found those mutations, we really were optimistic that a treatment would be developed. But the reason for that was because the mutations in BRAF switch it on and stick it in the on position. Now, there are other genes that get mutated, BRCA2, for example, where what the mutations do is they switch the gene off. BRCA2, both copies are switched off. Well, if it's already switched off, what do you do? And the gene is non-functional, the gene is damaged, you can't resurrect it, you can't switch it back on. And drugs don't tend to switch things back on anyway. Drugs are interferers. They're sort of crude things like, throwing an axe into an engine. That's what a drug is. It stops it working. So if you've got a gene that's already not working, what do you do? And that becomes a difficult problem for many genes across cancer.
Although we do have the very clever example of, you mentioned BRCA2, and then we have these genes called PARP inhibitors, which women who have breast and ovarian cancers with these mutations, I think men with prostate cancers as well now. So can you just explain how this works? So you've got the BRCA2 gene is inactivated, it's not working, but then we have drugs that work with them. What's going on there?
Yeah, so this is absolutely. So this is an example of what is called synthetic lethality. What the BRCA2 gene does is it confers, when it's mutated, both copies gone, the cell has this genomic chaos. But a cell is coping with that genomic chaos, just about. And there's a balance there. The genomic chaos is producing mutations. Those mutations allow the cells to become cancers, and that's why they come to our notice. But they're not very healthy cells despite that, because all those abnormalities, all that genomic chaos is not good for a cell. So the notion came, well, why don't we just increase the amount of chaos in those cells? Why don't we inhibit a different part of DNA repair so there's even more chaos in those cells? And that's what PARP inhibitors do. And the reason why they work so nicely is because in a cancer with both BRCA2 mutation, both BRCA2 copies gone, there is genomic chaos. You add more chaos in, the cells just can't cope and they start dying. But in the rest of the cells of that person's body, with one mutated copy, they're fine because they've got the normal copy, which is allowing them not to have genomic chaos. And so a little bit of PARP inhibition doesn't do very much to them. They survive that perfectly well.
It's a very clever sort of two wrongs making a right. We've got a question from Jane Cutler. Jane, I have to say, was the librarian of my village library when I was a child, and is partly responsible for my love of reading. So hello, Jane. Thanks for your question. She asks, I understand that breastfeeding can help to prevent breast cancer. If this is true, is it widely known? Sandra, you're nodding.
Yeah, yeah, that's totally true. That's one of the other examples that I missed in the previous questions. Yes, and this is actually one of the arguments that WHO uses to kind of promote breastfeeding. It is widely known for many of the epidemiological studies, and now this is something that we also have to continue promoting, because it's not only the benefit for the baby, but also for the mother.
I'm just scanning through our questions here. Picked some out. Here's an interesting question. Is cancer prevention a social issue? So I'm curious, is it sort of social as much as scientific? Many of the high risk behaviors can be linked to poverty. Sandra and also Maggie as well, I don't know if you have anything to chip in there.
Yeah, I think it's undeniable that socio-economical factors are important here. And obviously we touched a little bit on that before when we said about public health policies. It is true, and it's already recognized in many studies that the socio-economical factors are an important risk factor for development of cancer because of the behaviors that are associated to poverty. And we see inequalities in the weight, especially in the weight of cancer, not only the causes of cancer, but also the treatment. So yes, poverty is a big issue in cancer development, I would say, not only involved in causing cancer, but also involved in inequalities in treatment and treatment options for patients.
Maggie, do you have anything to chip in here?
No, other than that, I agree absolutely. It was something I was I've been thinking about more recently, and I suppose it was underlined by these sorts of points emerging from the death rates from COVID-19, that what became clear as things went on was the inequalities and the sort of poverty impacting much more heavily on certain groups and so on. And I think that equally applies to the health messaging about cancer and cancer prevention. It's fine to say, yes, we should all eat a healthy diet, have more fruit and vegetables, etc., etc. But that's the ability for some families to enact that can vary very significantly from others. Yeah.
It reminds me of the trip that we took to Kenya, because we went to Eldoret in Kenya to look at an area that had very high rates of esophageal cancer. And this is part of the Mutations project, trying to take samples and understand what's causing it. And we saw a man who'd been brought in and had come hundreds of kilometers in the back of a bumpy taxi because there's just no local healthcare. And it's really interesting to see that we could sort of forget that there are many, many, many people in the world, and their access to information and services is very different. Yeah. We have a question. I think I'll pop this one to you as well, Sandra. This is an interesting preventive one. In terms of preventative measures, how does the research on plant-based diets in reducing the risk of cancer weigh up? It's easy to make links with things like colorectal cancer, bowel cancer. Food goes through your bowel, obviously. But what about cancers outside the digestive tract? What can we say about particular patterns of diet and particular patterns of eating?
Yeah, well, I mean, many of the studies have also looked at other type of cancers besides colorectal cancer, and we see some interesting findings for other cancers. I mean, that is the question of nutrition and also obesity. Obesity is also, I mean, this is also related to nutrition, but it's also is one of the risk factors associated with other cancers like breast cancer, liver cancer, so esophageal adeno, for example. So there are, other cancers that are not directly in the digest system that could be affected by nutritional factors, by diet. And also, well, we know that a cancer is a disease it's a systemic disease, and also nutrition affects all the tissues of your body. So it affects the type of hormones that you're creating every single day. It also involves the microbiome, which is a completely different area of research, but it also involves, well, all the microbes that are working in your system and that help you to live normally. But it would be also affected by the type of food that you usually consume. So yes, the nutrition it's something that keeps us alive, but it's also something that it keeps all the tissues in our body alive, and that's why it affects directly any single organ, and obviously will have implications in cancer development in any single organ of our body.
So thanks very much. The next question we have is from Helen, and Mike, I'm going to put this one to you, and I wonder if like you and I might have slightly different opinions on this, but to what extent does the different extracellular environment or function of cells contribute to the incidence of cancer in different parts of the body? And, you know, our cells, no cell is an island, they're in tissues, they're in organs, they work together. So what do we know about the role of, you know, is it just about one cell doing its thing, or is it one cell as part of a tissue needs to go wrong in order to become a cancer? It needs to sort of, you know, rebel against the cells around it.
Well, I think that the tissue structure of different tissues, I think almost certainly is contributing to the differences in cancer risk in different tissues. And so a given mutation in a cell will have less chance in some tissues to expand and grow, and therefore go down the line to acquiring further mutations and becoming a cancer than it will in other tissues. And tissues differ so much in their structure that I'm pretty sure that those differences absolutely do contribute to the differences between tissues in cancer incidence.
And one of the things I'm very interested in is the role of inflammation in disrupting tissues and, you know, just making them all a bit so that cells can, you know, so if cells are a bit wrong, if they're in a bit wrong environment, they might be more likely to then develop into a cancer.
Yeah, absolutely. And that can happen a number of ways. Inflammation can increase the mutation load, and as you said earlier, keep your mutation load down. But it also, by changing the tissue structure, may allow cells to go beyond their normal confined regions easier in these damaged tissues than they would do within normal tissues. I think that's perfectly plausible.
Thank you. We've got a question from Humara. I'm going to put this to you, Sandra. How does ionizing radiation penetrate through cells and cause damage? So the question is about malignant melanomas, but I'm assuming there's different types of radiation we can be exposed to. There's UV radiation from the sun, and how is that different from ionizing radiation? What's the connection between these types of radiation and damage that can lead to cancer?
Both are different type of radiation, been like studied for a long time, but we know now that both radiations can cause mutations in cells, in mutations in DNA. They both have different effects on the DNA, and they have different profiles of alterations that they can cause. But
can I just jump in and clarify? So ionizing radiation, this is X-rays, basically, we're talking about. Yeah,
exactly. X-rays compared to UV light, which is a different kind of light. But as I said, I mean, the common thing about both exposures is that they both affect the DNA. They have the capacity to mutate DNA in a different way. They cause different alterations in the DNA. So because of and this is related to what Mike explained before, I mean, because of the type of alteration that is causing each of these radiations. The consequence, obviously, they both promote cancer in cells, but the consequence in all the mechanisms that go beyond that mutation and how this helps the cells to proliferate might be different and might differ in each cell type. So that's one thing. But something that we should remember is that, yeah, is known that both are carcinogens and both are mutagens.
So we need to watch out, don't add to your mutational burden. So just Maggie, just before we wrap up, I do want to come to you, you know, as a patient advocate, someone who runs a charity, listening to all this talk about science and research, you know, how important is it that patients are actually at the table when scientists like Mike and Sandra are deciding what questions we're going to be researching? If you could just sort of briefly talk to that.
Yes, it's very important because I think experiencing or having experience of the particular cancer can alter the relevance of the research question that's being contemplated by the scientists. It can alter if we're talking about clinical trials, can alter the design of the clinical trial, because what drives and infuses the scientists, the researchers, to examine and answer particular scientific questions doesn't always take account of how does that work when you're the patient. You know, there were certain kind of designs of trials where getting information requires lots of blood tests, for example. But if you're the patient being
you're a pincushion.
18 different blood tests in a short time, you will not see it in quite the same way. So those sorts of issues, I think, are acknowledged, and patient involvement right from the start is acknowledged now. It is growing in importance, and in the involvement of patients and carers at the very start of the whole sort of research process.
Thank you. I'm afraid we have to wrap up now. There's so many questions. I'm so sorry if we couldn't get to your questions, but thank you so much to everyone for engaging. I'm going to wrap up and hand back to Becky, but first, very quick plug for my book. Rebel Cell is available in paperback, out now from all good and all evil retailers, and it goes into a lot of detail about the genetics of cancer, how it evolves, a lot of the work that Mike's talked about that's been done at the Sanger, and the work that Sandra's doing at IARC talks about that, and what we can understand about what causes cancer, where's it going, and how we might actually use this knowledge to try and treat it more effectively in the future. So I'm going to wrap up and say thank you so much, Maggie, Mike, and Sandra, and hand back to Becky. Thank you, everyone.
Great, thank you, Kat. An excellent plug there. So a huge thank you to our chair, Dr. Kat Arney, for expertly guiding us through a subject that one way or another affects us all. Thank you again to our brilliant panel, Professor Sir Mike Stratton, Dr. Sandra Perez, and Maggie Blanks, for inspiring us, for all the work that they do, and I think giving us so much to think about, and I think that's come through with all the questions we've had this evening. I hope you've enjoyed it as much as we have. As is always the case, there are so many more questions than we could get through. Hopefully we've managed to cover a number of the themes that were coming through, but if you've got a burning question that we've not been able to answer, do email us and we'll endeavour to get an answer to you. We'll pop our address in the chat. We'll put links in the chat also to our website, where you can find out more about our future events, and also sign up to our newsletter to keep up to date with all the things that you can join in with. Hingston Hall Estate, Georgians, Jet Engines and Genes, a lot to get your teeth around there, launches in September and will be treating us to a guided tour through the historic 18th century buildings and extensive grounds at the heart of our home at the Wellcome Genome campus. You'll also find links in the chat to useful resources connected to our panel and the discussions from this evening. And if you found yourself affected by the themes of tonight's talk, there are also some links to places where you can find out more information and get some support. If you'd like to watch this event again or any other recordings from the 2021 Genome Late Season, or share them with your friends, please go to our Genome Gallery events page. As we close tonight, we'll be posting a short survey. I know everyone asks these, but please do, if you can, stay online. They're really helpful for us to have your feedback, and it helps us shape our future event programming. So just finally, thank you again to all of our participants and to all of you for joining. Take care, enjoy the rest of your evening, and we'll see you again soon. Good night.